Hello everyone, very good evening. Welcome back to my YouTube channel. In today's lecture, we will be talking about glycolysis. The word glycolysis can be split up into glycus and lysis. Glycus means sweet and lysis means breakdown. As this pathway was discovered by Emden, Meyerhoff and Parnas, it is also called as EMP pathway. The site of glycolysis is cytosol and there are total 10 reactions in glycolysis. It is a central universal pathway which occurs in all the organisms. It is a fundamental energy generating pathway. During this pathway, partial oxidation occurs, as during complete oxidation, H plus ions, electrons and carbon dioxide is released. But during glycolysis, CO2 is not released. Thus, it is said that during glycolysis, partial oxidation occurs. It is an oxygen independent pathway. Even if oxygen is not present, it will occur. During glycolysis, one molecule of glucose, which is a 6-carbon compound, gets converted into 2 molecules of pyruvate, which is of 3 carbon. Net gain of energy is 2 molecules of ATP and 2 molecules of NADH. And reaction number 1, 3 and 10 of glycolysis pathway are irreversible. During glycolysis, substrate level of phosphorylation will occur, during which ATP is synthesized, and oxidation will occur, during which NADH plus H plus is synthesized. So these 10-step reactions can be divided into preparatory phase and payoff phase. So during preparatory phase, energy is utilized, and during this, one molecule of glucose gets converted into 2 molecules of glyceraldehyde 3-phosphate. During payoff phase, energy is generated and 2 molecules of glyceraldehyde 3-phosphate gets converted into 2 molecules of pyruvate. So let us look at this 10-step reaction and then we can study each of them one by one. So first, in the first reaction, one molecule of glucose gets phosphorylated into glucose 6-phosphate with the help of enzyme hexokinase, and during this reaction, one molecule of ATP is utilized. In the next step, glucose 6-phosphate gets isomerized into fructose 6-phosphate with the help of enzyme phosphofructose isomerase. In the next step, fructose 6-phosphate again gets phosphorylated into fructose 1,6-bisphosphate. During this reaction, second molecule of ATP is utilized, and the enzyme which is involved in this reaction is phosphofructokinase 1. So till glucose to fructose 1,6-bisphosphate, 2 molecules of ATP are utilized. In the next reaction, fructose 1,6-bisphosphate splits up into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which are of 3 carbon. As glycolysis proceeds with glyceraldehyde 3-phosphate, this dihydroxyacetone phosphate gets converted into glyceraldehyde 3-phosphate with the help of enzyme triose phosphate isomerase. Further, glyceraldehyde 3-phosphate gets converted into 1,3-bisphosphoglycerate by utilizing pyrophosphate, and the enzyme which is involved in this reaction is glyceraldehyde 3-phosphate isomerase. During this reaction, NAD plus gets converted into NADH plus H plus. So now in this reaction we have 2 molecules of NADH plus H plus. As now we are proceeding with 2 molecules of glyceraldehyde 3-phosphate. Now further, this 1,3-bisphosphoglycerate gets converted into 3-phosphoglycerate by synthesizing 2 molecules of ATP. And the enzyme which is involved in this reaction is phosphoglycerate kinase. Again, this 3-phosphoglycerate gets converted into 2-phosphoglycerate with the help of enzyme phosphoglycerate mutase. 2-phosphoglycerate gets converted into phosphoenol pyruvate by releasing one water molecule, and the enzyme involved is inulase. Then phosphoenol pyruvate gets converted into pyruvate with the help of enzyme pyruvate kinase, and again during this reaction 2 molecules of ATP are synthesized. Now this is synthesized. This is again synthesized. This is synthesized. So net gain is total 2 molecules of NADH, or you can say total molecules synthesized are 4 ATP and 2 NADH plus H plus. But in the initial reaction we have utilized 2 molecules of ATP, thus net gain is 2 molecules of NADH plus H plus 2 molecules of ATP. We will see these again when we will study each reaction one by one. This is just for your information. Okay, so just have a look at these reactions. If you have any doubt, any query, you will understand everything when we will study each step one by one. Okay, now let us move further. Let us study each reaction in detail. Now reaction number 1. This first reaction, as we have said in the introduction part, reaction number 1 is irreversible reaction, and during this reaction glucose, which is a 6 carbon, gets converted into glucose 6 phosphate, and the phosphate which is released from ATP is attached to carbon number 6 of glucose, and the enzyme which is involved in this reaction is hexokinase. So this hexokinase requires ATP in the form of Mg2 plus ATP complex. This Mg2 plus positively charged Mg2 plus ions bind to negatively charged phosphate ion, and then the gamma phosphate is transferred to carbon number 6 of glucose. So this hexokinase requires ATP in Mg2 plus ATP form. Now let us study few things about this hexokinase. There are 4 types of hexokinases. So basically this hexokinase, it is a non-specific enzyme. It is a non-specific enzyme. It can add phosphate to any 6 carbon sugar. It can add phosphate to any 6 carbon sugar. Now as we have said, there are 4 types of hexokinases: type 1, type 2, type 3 and type 4. So type 4 is glucokinase, which is specific for glucose, which adds phosphate to glucose molecule, and it is present in hepatocytes. Now inhibitors and activators of hexokinases are. So ATP, which is present without Mg2 plus, is an inhibitor of hexokinase, as well as glucose 6 phosphate, which is when present in excess amount can also act as an inhibitor of hexokinase. AMP and ADP, which defines or which suggests energy deficient condition, are activators of your hexokinase. Now in the reaction 2, glucose 6 phosphate gets converted into fructose 6 phosphate with the help of enzyme phosphohexosisomerase. It is a simple conversion of aldose to ketose. In reaction 3, fructose 6 phosphate gets converted into fructose 1,6 bisphosphate. During this reaction, phosphate from ATP is transferred to carbon number 6 of fructose, and this reaction occurs with the help of enzyme phosphofructokinase 1, that is PFK1. This enzyme also requires Mg2 plus ion. Now this reaction is also an irreversible reaction, and it is a first committed step of glycolysis. Why so? So the glucose 6 phosphate, fructose 6 phosphate, which are synthesized during an earlier reaction, can also enter other metabolic pathways. But once fructose 1,6 phosphate is synthesized, it is confirmed that glycolysis will occur. Now this reaction is a first committed step of glycolysis. Some bacteria, protists, and all plants use pyrophosphate instead of ATP. Now this phosphofructokinase 1 is a tetrameric enzyme, and each subunit has 2 ATP binding sites. So first site is for inhibitory ATP binding and second site is for substrate ATP binding. So if one ATP molecule is bind to one subunit, next one will come and it will bind to another subunit. Third one will come, it will bind to third subunit. Fourth one will come and it will bind to fourth subunit. So you can also say that this ATP is an allosteric inhibitor of your enzyme phosphofructokinase 1. Now citrate, high proton concentration, are inhibitors of phosphofructokinase 1, and ADP or AMP, ribulose 5 phosphate, which is an indirect activator, or you can say fructose 2,6 bisphosphate is an activator of phosphofructokinase 1. Now let us see few points about this fructose 2,6 bisphosphate, how it activates your enzyme phosphofructokinase 1. So during glycolysis, glucose gets converted into glucose 6 phosphate, which further gets isomerized into fructose 6 phosphate, and when this fructose 6 phosphate gets converted into fructose 1,6 bisphosphate, the enzyme PFK1 plays an important role. But sometimes this fructose 6 phosphate gets converted into fructose 2,6 bisphosphate with the help of enzyme phosphofructokinase 2, and this synthesized fructose 2,6 bisphosphate will activate phosphofructokinase 1, which then will synthesize more fructose 1,6 bisphosphate. Thus it is an activator of PFK1. This fructose 2,6 bisphosphate is an activator of PFK1. But sometimes when this phosphofructokinase 2 gets phosphorylated. When this phosphofructokinase 2 gets phosphorylated, it behaves like a phosphatase, and it is also called as fructose 2,6 bisphosphatase. This phosphorylated phosphofructokinase 2 will convert fructose 2,6 bisphosphate to fructose 6 phosphate. So in this case, fructose 1,6 bisphosphate won't be synthesized. I hope you have got this point. Now in the next reaction, that is reaction number 4, fructose 1,6 bisphosphate, which is a 6 carbon compound, gets splits up into 2 3 carbon compound. First one is dihydroxyacetone phosphate and second one is glyceraldehyde 3 phosphate. Carbon number 1, 2 and 3 of your fructose 6 phosphate, these carbons, gets converted into dihydroxyacetone phosphate, and carbon number 4, 5 and 6 gets converted into glyceraldehyde 3 phosphate. So please remember this, it will come in your exams. Now the enzyme which is involved in this reaction is aldolase. There are two classes of aldolases. Class 1 is usually observed in animals and plants, and class 2 is found in fungi and bacteria. As glycolysis proceeds with glyceraldehyde 3 phosphate, ultimately this dihydroxyacetone phosphate gets converted into glyceraldehyde 3 phosphate with the help of enzyme triose phosphate isomerase. So now ultimately we have two molecules of glyceraldehyde 3 phosphate which will enter in further reactions. Now in the reaction number 6, glyceraldehyde 3 phosphate gets converted into 1,3-bisphosphoglycerate with the help of enzyme glyceraldehyde 3 phosphate dehydrogenase, and the phosphate which is attached to carbon number 1 is taken from pyrophosphate. Now during this reaction, NAD plus gets converted into NADH plus H plus. Now there are two inhibitors of this enzyme glyceraldehyde 3 phosphate dehydrogenase. First one is iodoacetate and second one is arsenate, which is an analog of phosphate. So you can see the structure of arsenate, it is AsO4 3- and phosphate is PO4 3-. Now let us see what will happen if arsenate is attached instead of your phosphate. During glycolysis, as we have seen that glucose gets converted into glyceraldehyde 3 phosphate, and when phosphate is attached from pyrophosphate molecule, this glyceraldehyde 3 phosphate gets converted into 1,3-bisphosphoglycerate. When this 1,3-bisphosphoglycerate gets converted into 3-phosphoglycerate, one molecule of ATP is synthesized, which we will see in further reactions, and ultimately this 3-phosphoglycerate gets converted into 2-phosphoglycerate and blah blah blah. Ultimately we will get pyruvate. But if instead of phosphate arsenate is added, this glyceraldehyde 3 phosphate gets converted into 1-arsino 3-phosphoglycerate. Now if arsenate is attached, ADP will not be synthesized. You will not get ADP, as this arsenate will be transferred or released while conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. So ultimately no ATP synthesis in if this arsenate is added. Now as we have seen in earlier session that in glycolysis, two molecules were utilized in the initial reaction, and 4 were synthesized and 2 molecules of NADH were synthesized. But as in this reaction we have seen that if arsenate is added, only two molecules will be synthesized while conversion of phosphoenol pyruvate to pyruvate. But in the initial reaction we have utilized two are synthesized and two molecules of NADH are already we have synthesized. Now when we will remove this two utilized and two synthesized, we have 0 ATP synthesis. So if arsenate is added as an inhibitor during glycolysis, we have got only two molecules of NADH. Now I hope you have got this point. If not, you can ask me in comment section. I will explain you there, or else I will put a short video on that. Then you will get it. Now in the reaction number 7, 1,3-bis phosphoglycerate gets converted into 3-phosphoglycerate. Phosphate molecule from the carbon number 1 is transferred to ADP and ATP is synthesized. This reaction also requires phosphoglycerate kinase, and now from this we have got two molecules of ATP. In the reaction number 8, 3-phosphoglycerate gets converted into 2-phosphoglycerate with the help of enzyme phosphoglycerate mutase, which transfers a phosphate molecule within the group, or like mutase transfers the phosphate within the molecule. So this enzyme also requires Mg2 plus ions. In the next reaction, that is reaction number 9, 2-phosphoglycerate gets converted into phosphoenol pyruvate with the help of enzyme enolase. During this reaction, one molecule of water is released, and the inhibitor of this enolase is fluoride. Inhibitor of this enolase is fluoride. This fluoride is usually present in toothpaste. Now, if this fluoride will inhibit enolase, it will ultimately prevent the glycolysis in bacteria. Okay, so this is the inhibitor. And in reaction number 10, which is the last reaction, this phosphoenol pyruvate gets converted into pyruvate, and during this reaction phosphate from this carbon number 2 is transferred to ADP, and ADP gets converted into ATP. Now again we have two molecules of ATP which are synthesized. The reaction occurs with the help of enzyme pyruvate kinase, and this enzyme also requires Mg2 plus ion. The inhibitors of these enzymes are ATP and alanine, which is an amino acid, and activators are fructose 1,6-bisphosphate. When fructose 1,6-bisphosphate is synthesized, we all know that it is a confirmation that glycolysis will occur. Thus, it is an activator of your fructose 1,6-bisphosphate. Now, further, during aerobic reaction, this pyruvate will enter TCA cycle, which will then enter ETC, and energy is synthesized. But when it enters an aerobic reaction, or when oxygen is absent, this pyruvate gets converted either into ethanol and during this reaction NADH is utilized. We will see this in feat of pyruvate. I'll post one more video on that, so in that you will get each and every reaction in detail, what happens with the pyruvate. Now again we will see net gain of energy during glycolysis. Now during conversion of glucose to glucose 6 phosphate, we were utilizing one molecule of ATP. Fructose 6 phosphate to fructose 1,6 bis phosphate. We were utilizing one molecule of ATP. Then during conversion of glyceraldehyde 3 phosphate to 1,3 bis phosphoglycerate, we had synthesized 2 molecules of NADH plus H plus. These were utilized and this is synthesized. While conversion of 1,3 bis phosphoglycerate to 3 phosphoglycerate, we had synthesized 2 molecules of ATP, and conversion of phosphoenol pyruvate to pyruvate, we had synthesized 2 molecules of ATP. Now these were total 4 and these are synthesized. Now when we will calculate net gain of energy, we have ultimately got 2 molecules of ATP and 2 molecules of NADH. This 2 molecules of NADH corresponds to 5 ATP, as this one NADH corresponds to 2.5 ATP. 2 molecules will give you 5 ATP, and we already have synthesized 2 molecules of ATP during substrate level phosphorylation reaction. Thus, net gain of ATP or energy during glycolysis is 7 ATP molecules. So I hope you have got all the steps and all the reactions which I have taught you. So see you in the next lecture. Till then, bye bye. Thank you so much. If you have any doubt, any confusion, you can ask me in comment section. I can answer you over there, or else I can post some short video regarding that, and then maybe we can have a little discussion regarding next topic. If you want me to teach any particular topic, do let me know. And thank you so much for patiently listening my lecture. Thank you so much. Bye bye.